Yeah, 60.
Okay, so I'll get a syllabus up on the website, and you can look at that. It's the same stuff as last class. If you haven't looked at if you're new to the class, Kenna, you can look over the syllabus and get back to me. I get bored by doing the syllabus, and I know you guys get bored by it, so we're just gonna move on. What are we doing this week? We've got lab on Wednesday. That you would have seen that on the syllabus, but I didn't print it. So we got lab on Wednesday. It's going to be a computer lab. So bring your laptop. How many people don't have a laptop? Or actually, I'm not going to call people out. Email me if you don't have a laptop and you need me to bring a laptop. Okay. So, but we're going to be on laptops. We're going to be doing computer stuff to start out here. So this lab this semester is one giant laboratory activity. So last lab was like half of that the lactate dehydrogenase isolation. You're going to take an enzyme and you're going to change an amino acid in it, and then you're going to do kinetics on it to study it. So that's going to be the whole lab outside of an extra credit lab at the end. On Wednesday, we're going to start by picking our amino acids. Okay, so I'm going to want you to bring your computer, and we're going to download a piece of software called PyMol on it, and we'll do that. I'll get some directions out to you guys. We're going to have a quiz on Friday.
This Friday?
Yes.
Say that on the
So we have a quiz on Friday. We will have a help session. Help session on Thursday. And that's 8:30. Whoops. 8:30 to 9:30 p.m. on Google Meet. And the link for that's in your syllabus. It's the same link actually as last time, but I'll get the syllabus up on the website. You can look for it there. Yes.
Is the quiz like on stuff we learned this week, or
Yes.
is it like review?
It's like this today and tomorrow, or Wednesday. Yeah.
Okay, that's
So help out. That'll be Google Meet, and then my office hours this semester are a little different. Last time I did this thing by appointment, and I was always kind of in my office anyway, so I'm not going to worry about trying to leave things open if people aren't coming. So office hours are going to be 1:15, so right after this class, p.m. to 3:15 on Mondays, and then 1:15 p.m. to 2:15 p.m. on Fridays, or by appointment. So if you need to see me outside of those times, let me know. We'll see if we can work out a mutual time that works for everybody. But like I'm happy to see people outside of those times. So let me know either by email or just tell me after class. Hey, talk to me after class about when we can set up a time to talk. Questions on that stuff? Yeah.
Is there a pre-lab Wednesday?
There's not a pre-lab. There's no way I'm going to get that together in time. Okay, I was probably as disappointed that that lab was on the syllabus as you were. I'd forgotten that we do it first week. But I remember now, you know, one first week we had a snowstorm, and I said, "You guys are all on campus, come anyway." So, and then anybody who was off campus, I'd let do something different.
How were your reviews? Class evaluations?
I don't remember. We have to do the lab anyway. So anyway, so all right. So we're going to start by reviewing what we've got here in terms of energy, right? Okay. So when we think about biochemistry, well, actually, let's talk about this semester versus last semester first. So this semester versus last semester. Last semester was all about structure and function. Yes. Last semester was about structure and function of our four major classes of molecules. Okay, I'm recording, you just can't see it.
Oh, okay.
So What are our four major classes of molecules that we did structure and function of? Lipids. Okay, give me another one.
Carbohydrates.
Carbohydrates. Proteins.
Nucleic acid.
And nucleic acids, right. All right, those were our big molecules. That's what we talked about last semester. This semester, we're going to talk about energy flow. Most of it is. There's some nucleic acid stuff at the end. Okay, so in other words, we're going to be talking about how food goes to ATP. Okay, that's, I mean, more than that, but that's going to be a lot of what we're talking about. All right, so to review some things we talked about at the end of last term, we looked at this. We looked at how we can break metabolism into two different sections. We can talk about catabolism and anabolism. And so, did I say it wrong? Oh, and anabolism, thank you. All right, and this chart kind of shows what's going on with these things. All right, so if we wanted to think about characteristics of, let's say, catabolism, we can look at our chart. Hopefully you guys can see it okay. I can't really make it bigger without losing it. Actually, I could do this. I don't know whether I'll be able to write, but we'll try it. Okay. So we've got catabolism. If we look at that, that's right here. We take complex molecules and we break them down into simpler molecules, right? So from the perspective of oxidation and reduction, what's going on there? Our complex molecules are being oxidized. And what's happening? Well, actually, let's do it in a separate category. Complex molecules are being oxidized. The counter to that is we have something oxidized, something's reduced. What gets reduced?
Energy.
Yeah, energy cofactors get reduced. Okay. Right. We're gonna talk about this being convergent. Okay, so we're going from a lot of different things down to a very small number of molecules. Okay. The process is convergent.
How is it going down into a smaller amount?
A smaller number of kinds of molecules.
Oh, okay.
So what we're doing is, I mean, these are completely depleted products, right? But we could go down to these. We could take these carbohydrates, fats, and proteins, and if we completely oxidize them, we're going to make them into carbon dioxide and water and ammonia, right? Completely break them down. Not all those products are fully oxidized, but
so it's like less complexness almost.
less complex. So what's going on is, well, here, I'll show you the next figure. The next figure is good for this. So if we talk about catabolism, you've got all these different types of molecules. These are just some of them. And you're going to try and break them down into simpler and simpler molecules until you're going to break them down into acetyl-CoA if possible. All right. And the acetyl-CoA can go through the citric acid cycle, which will then completely oxidize them down to carbon dioxide. Okay. So you're looking, catabolism is a process of trying to you try to, well, it's not you, and evolution isn't thinking about it, right? It just happened because it's efficient. But it's efficient to take a large number of things and try to get them into a common set of pathways as soon as possible. So by making this large number of molecules into a common set of molecules, then you can not have as many pathways. I'll look at my notes because I feel like I'm forgetting one here. Might be it. No, that's it. Okay. All right, so anabolism is the opposite. All right, we've got simple molecules built up into complex molecules. Okay, if the simple molecules are being oxidized or reduced. Okay, as we build them up, those complex molecules are more reduced overall than the simple molecules that went into them. If we're going to reduce those, we need to oxidize something. What are we going to oxidize? Yep, energy cofactors are oxidized. Okay, and the process is divergent. All right, you take a small number of simple building blocks and you build them up into many, many different molecules. All right. One easy way to think about that with what we know is you have 20 common amino acids, right? And you make them up into 30,000 different proteins. Questions? Just kind of in terms of thinking about that. Okay, and then I like this figure. We've already used it. But you can see from the divergent side that we'll start with acetyl-CoA and maybe some other things. Glucose is often used. It's not in this figure, okay? But acetyl-CoA and other things, and we can build up a tremendous number of different molecules. And we're going to talk about both parts of this. 2/3 of the semester will be about this right here, and this. But and then the last bit we'll talk about what goes on with nucleic acids and making proteins and such. It'll actually be, is anybody in molecular biology right now? Is that this year? Okay, there's a lot of overlap with molecular biology. All right, let's talk about what some of those energy cofactors are. So we did this last time, but these are the energy cofactors we're going to need to know. So this is NADH, okay, NAD plus and NADH. And like last semester, you still need to be able to identify this, name it, talk about whether it's oxidized or reduced, and draw the other form. So name, oxidized or reduced, draw the other form. Okay, and you need to draw the other form with all atoms, but of course you'd be given one of them, so you just need to be able to show it different, and you can use R's and things like that. All right, and then this was for this, I don't want you to focus, so we're looking at NADH. We don't want to focus on any stereochemistry, so all we care about is one side of this, right? We're not going to worry about there is no real stereochemistry here, although if you had one of these radioactively labeled, you could tell the difference. But what we've got is this right here. This is NAD plus, right? And the difference is this gets reduced if you add a hydrogen to it and you oxidize it, you take electrons away. I'm sorry, you reduce it. You don't take electrons away, right? We have a positive charge. We're going to bring in electrons. We're going to pop these here, pop these on top of there. We end up with this is what our reduced form looks like. So it's all this other stuff plus that. Okay? And we also talked about, my daughter's maybe going to do a science project that involves adenosine. So I got to look back in my backpack textbook I used to use, where they talk about how this is a really good handle that gets used over and over again. Remember, we've got that ADP is part of this. It's not part of it that does anything. It's not functional. This has the H on it as which could be donated to things as a hydride, right? This part. This has been kind of conserved by nature because it's a great thing for enzymes to grab onto. And if we come down here and we look at FAD, its reduced form is FADH2, we see that same thing again, okay? I'm once again going to cross off a piece. This is the 1 electron form. We're just going to do all we're going to do is 2 electron transitions in this course, right? Things are going to get 2 electrons always, and they're going to give up 2 electrons. So if we take FAD, this is our business end of FAD right here at the end. It can accept an hydrogen here and a hydrogen there. 1 electron comes with each, and we end up with a reduced form. Okay, so you can see we end up with more electrons here than there. All right, when I think about this, if somebody was to give me this and I was to try to remember how I put how this worked out with the bonds, I would do it like one of, so let's go ahead and draw. This is how I do it when I'm trying to remember it. I don't have the structure drawn, so I'd have to be relying on getting one of these, recognizing it, right? You don't have to draw it from scratch. And I don't know if this helps for you guys. You'll have to let me know. So let's say I have the oxidized form. Okay, so what I've done is I've taken this, I've got an R here. When I think about it, each hydrogen brings 1 electron, right? So I could think of trying to go from one of these to the other. I could think about, I've got my hydrogen here, right? It's going to donate electron. This is one of those 1 electron arrows. Donate an electron. 1 electron goes there. 1 electron comes here. 1 electron from here. It's a 1 electron arrow. 1 electron comes here to put the H on. I don't know if that helps or not. This would be contributing 1 electron as well. But that's how I would go from one to the other. And then from that, I would get to that. But you need to, once again, you need to know, be able to draw it, not draw it from scratch, recognize it, name which one it is, name it, right? Is it the oxidized or reduced form? And draw the other form. Okay. And then once again, here's our nice little ADP, right? Conserved again. We haven't done, so in terms of that conservation, we haven't done acetyl-CoA yet. We will. Okay, we're not going to do it today. But acetyl-CoA also, ADP's in it. Okay, just over and over again, ADP got used by evolution as a great handle for things. Questions before I move on from cofactors? Actually, I'm going to talk about ATP real quick. Okay, so ATP you should be able to draw from scratch. It's super important, right? Okay, so you need to be able to draw all forms of ATP from scratch. So ATP, ADP, or AMP from scratch. Of course, you need to be able to recognize them as well. So I'll do that here. may not have left myself enough room. I don't know that I would make a big deal about whether you've got the DNA form or the RNA form. The RNA form is really, when you think about ATP, right, the energy source, molecule gets used over and over again, it's going to be the ribo form, or it's the RNA form. Oh, and I was complaining. I was in with my Gen Chem students today, and I had to draw them a molecule I was trying to draw a fatty acid to explain how London forces add up, and it was killing me. I had to draw every atom out because it's Gen Chem. I need some more room here. It was probably killing them too, watching me draw stuff. Okay, so there's ATP. And so ATP, if ADP were here, right? We have AMP were here. Questions? Okay. All right, so then ATP is used you couple the hydrolysis of ATP to reactions, right? And it's coupled to reactions to make them spontaneous. So what we think about if we were to hydrolyze ATP, well, there's all sorts of ways you can hydrolyze ATP. We're going to talk mostly about, right now, about the hydrolysis of ATP to ADP. So I'm going to write it down here at the bottom. We'll move it up. But ATP plus water going to ADP plus, I'm just going to write the phosphate like that. Okay. But you should be comfortable doing this kind of all atoms. And so we could imagine if we're going to hydrolyze this, that we would have, I'm going to do it with water, but I'm actually going to do it with hydroxyl. Then I don't have to do multiple steps or deprotonate afterwards. You guys okay with that? All right. So I'm going to go ahead, and which of these would I need to hit? Which phosphate would I need to hit to make this into ADP? Okay, so I could hit here, right, and I could pop up this way. You could also hit here and pop that way, right? Either of those. Actually, off the top of my head, not sure which one it is, usually. But so if I went to here, then I would need to pop this here, right, to make it. Mostly, I'd be concerned that if you were to draw something, that it would actually be reasonable. And it may be one of those is more common than the other, or they might both happen. Okay. Sometimes things are driven by, you make an AMP, right? When you think about adding or polymerizing DNA, right, or RNA, you take an ATP and you pop off the pyrophosphate, right? So that nucleophilic attack is happening here. Okay. And it has to happen here because this is what gets attached. We're not going to worry. We'll worry about that mechanism in a couple months. Okay. Usually the ATP is a direct participant in the reaction. Okay. So in other words, it's going to react directly with whatever our substrate is, and it's providing a leaving group. Okay, it's going to provide a phosphate, or it might provide an AMP. I'm trying to think of a case where you get a pyrophosphate that gets added. I don't have one off the top of my head. Nature's pretty diverse. I expect it happens. So what we have here is it's not that you have ATP is hydrolyzed over here and you have your reaction over here and somehow that energy gets transferred. Sometimes you actually end up with things like that, right? Okay. Sometimes you end up with weird things where a phosphate gets added and the charge causes a change in protein conformation and you actually have the reaction happen. But usually what's going on is something like this. So this is an example of glutamate can spontaneously, well, actually glutamine, I should say, spontaneously oxidizes to make glutamate. Okay, and you need to have the correct ratios of these. Both of these are actually neurotransmitters, as well as being components of components of proteins. So you have to have the right ratios of these in the body. And so the glutamine needs to be regenerated. So we have glutamate, and what happens is the ATP here is actually we're going to end up with a nucleophilic attack from here onto the ATP. We're going to end up with a phosphate on the end of this carboxylic acid, right? And then that phosphate can be displaced by an amine to make the amide. So let's see. Certainly here I have enough detail without redrawing whole ATP. We could have this happen, right? And it would look like this if I had it in one step. I don't know if the mechanism actually involves it kicking up and kicking down and then kicking out. But you see how we have it's participating here? Otherwise, this reaction is difficult to pull off because then otherwise the leaving group, instead of being a phosphate, which is very stable in solution, the leaving group would have to be an O2 minus. Questions? Yeah.
So, is that like how ATP usually works? Like it just like provides a leaving group, like that's why it's so powerful?
Yes,
And
it's gonna provide a leaving group in a lot of situations. Not all situations. So do you remember the sodium potassium ATP transporter? So it has or ATPase, I should say. So it gets a phosphate stuck on it, and it's very negatively charged, right? That causes it to change shape, and then the phosphate gets hydrolyzed off, and it changes back. So its shape change is being driven by adding a phosphate, and that's about charges.
Okay. Gotcha.
And then in your muscles is kind of separated more. Okay, it's probably something like the shape change, but you kind of have ATP is hydrolyzed, and the muscle fiber changes shape based on that. It's not a leaving group situation. Okay, so another thing to think about here in terms of these phosphates is phosphates can be spontaneously transferred from higher energy compounds. And when I say higher energy, I mean the energy of hydrolysis. So in other words, when we look at this delta G of hydrolysis over here, what we're saying is if we were to take phosphoenolpyruvate over here, so the top one, this value right here of about negative 65 kcals per mole is, I'm going to do phosphoenolpyruvate, and we'll put the phosphate on it. Okay, or let's do it like this. So phenylpyruvate, whatever pyruvate, and that's going to, if we take the phosphate off of that, let's do creatine. We've done CR for creatine before. That's easier. So if I have creatine phosphate, right? If I add water to that, without drawing the whole structure of creatine here, I'm going to be lazy. We get creatine, and then we get phosphate separated, right? That's the hydrolysis. What this is saying is that the energy of that is somewhere in there. I thought it was about 45, but I don't know. We'll say approximately negative 47 looking at this graph, right? Kcals, no, I'm sorry, kilojoules per mole. Does that make sense of what this chart's about? So we can look at the energy. If I hydrolyze off this phosphate, it's not that much energy. If I hydrolyze off the phosphate off a creatine, it's about negative 47 kilojoules per mole. This phosphoglycerate, it looks like it's about, I don't know, do I measure from the phosphate? Where do I measure from? Anyway, negative 50, right? Where do I read that over? But what they're trying to show you here is that, I guess I measure from the phosphate, that what you can do is you can take these higher energy phosphates and you could transfer them to another molecule. So if I have this without a phosphate, this could spontaneously be transferred there, could then be spontaneously transferred there, then down to here, then down there, etc. So you can take these phosphates until we finally end up with the energy of a free phosphate. All right, so those are energies of hydrolysis they're using, but you can think about, you can transfer those phosphates from higher energy to lower energy spontaneously. If I do that, I'm going to release energy, right? And if we're talking about a metabolic pathway, or talking about a catabolic pathway, hopefully you would capture most of that energy with some sort of energy cofactor. Okay, you either make ATP, you make NADH, you make FADH2. That's hopefully what's going to happen over that process. So let me write that. So phosphates are spontaneously transferred, or can be spontaneously transferred, from higher energy molecules to lower energy molecules. And that's what that figure is about. Questions on that? Just thinking about that? I want to make it clear, when I talk about something being spontaneous, well, let me ask you, why do you just think for a minute, when I talk about something being spontaneous, what do you think I mean? The reaction can happen.
There's no energy required.
There's no energy required, energy to be released, right? What about the speed of the reaction?
It doesn't matter.
Nothing. I'm saying nothing about how fast it happens. Okay, so it might never happen, right? Or might not happen on the range of on the lifetime of the universe, but it would release energy if it happened, right? So we might need an enzyme to help it go.
Well, it was 25 where they categorize high energy to low energy compounds.
Because it's, so what, I'll speculate why the authors, this is a picture from a book that I think is probably not your book, but this is why I think the authors did this categorization here is because ATP, we usually think about ATP as a high energy compound, and then these are compounds that ATP is used to add phosphates to. So in glycolysis, you're going to take glucose and you're going to make glucose 6-phosphate as one of those steps. So it's ATP is probably our line. Okay, since we're talking about thermodynamics and spontaneity here, let's just review quickly how we think about these things, or the equations we use. I'm going to remind you that we have this equation for the free energy, right? The actual free energy. So the actual free energy under biochemical conditions, well, we're not going to worry about conditions for the actual free energy, is equal to standard free energy under biochemical conditions plus RT, a correction factor, ln of Q, where Q is the reaction quotient. So let's say our reaction was A plus B goes to, or is in equilibrium with C plus D. Then Q would be products over reactants, so the concentration of C over the molar concentration, or times the molar concentration of D, divided by the molar concentration of A over the molar concentration of B, like that. All right. And so what we can see in this, if we think about this reaction, this helps out with our Le Chatelier's principle. We can use our Le Chatelier's principle, or we can relate our Le Chatelier's principle to this. So let's say the reaction quotient is whatever, okay? If I take D and I make the concentration of D much smaller, right? If I take D and I make it much smaller, then Q becomes, what happens to Q?
It's smaller.
This becomes smaller, right? And a smaller number here reduces this term. What happens when the top is less than the bottom? Well, with this, let's say make this smaller enough that this is less than 1, right? What's the natural logarithm of something less than 1? It's negative, right? It's negative, which makes this term negative, which makes this more spontaneous, right? So what that's telling us is as we reduce, what the math tells us is as we reduce the concentration of these products, the reaction becomes more spontaneous. Yes? You hear that? And what does Le Chatelier's principle tell us? If I pull these out, I move the reaction this way. Okay. So all these things agree. And I recommend that, when you're trying to think about things, if you don't have to mathematically quantitate it, use Le Chatelier's principle left, right, and center. Okay. It's one of our big things. It's not going away because we're going to talk about a lot of reactions this term. All right, and so this actually becomes important for reaction chains, and we did this a little bit last semester when we talked about the bicarbonate buffer system. But imagine, so we're going to have a bunch of long reaction chains we're going to talk about. Imagine we have A goes to B, that's an equilibrium, and B goes to C, and that's an equilibrium, and then C goes to D. And I've drawn these in a particular way. So let's say the under whatever conditions we're at. So I'm going to do this as real free energy, not standard free energy. What's the difference again? What is standard? When we think about concentrations, what are concentrations in standard conditions? 1 molar everything, right? Okay, this never happens in a cell. So let's say these are our real concentrations in the cell. Let's say this is about 0, okay? The free energy of this reaction is also about 0. And then let's say the free energy of this reaction is much, much less than 0. Well, what's going to happen here? This is a very spontaneous reaction, right? So if we assume that the kinetics are right, that it happens, and we're going to do that. Okay, we're going to assume there are enzymes around to make these things happen. The concentration of this is going to be really low, right? Or actually, the concentration that's going to be really high. Sorry. The concentration of this is going to be really low because we're going to pull it all through to make D. Do you agree with me? Save a little. So this is very spontaneous, right? This is very spontaneous. So we're going to make lots of that. Every time we make a C, we're going to go ahead and make a D, assuming that kinetics are good, right? So this is very low, so what's going to happen to this? If C is very low, what happens to my equilibrium between B and C? It's going to go this way. Because B is going to go to make C because C is really low. Now B is really low, so A is going to go and make B because B is really low. So you can have a chain of reactions here where these are kind of barely spontaneous or barely not spontaneous. They're kind of close to the line. And you can pull all these things along by having something at the end that's very spontaneous. Okay, or you could pull them along. I mean, you could then use D for something. And we actually saw this last semester in terms of one reaction. We saw it more than one, probably. But do you remember? Well, I mean, we can just use, if we talk about DNA. What was that?
Was the carbon dioxide
Carbon dioxide with?
ocean?
We did have the bicarbonate buffer system. We did have places where this sort of thing pulled along. Yes, that wasn't where I was headed, but I agree. That's an example of where you had multiple reactions and you could pull from one side to the other, right? Or actually, while we're talking about it, let's think about, rather than the ocean, this is our bicarbonate buffer system. I don't know why I froze up on that. Okay, so, all right, so we've got the bicarbonate buffer system. Why is the pH in my lungs higher than in my tissues? Is oxygen in this? No. What is it?
More carbon dioxide.
Less carbon dioxide, right? In my lungs, I'm exhaling carbon dioxide. All right, I exhale carbon dioxide at 4%, and I breathe it in at about 0.04%? 400 parts per million, 0.4%, not sure which. You guys can do the math. But at 400 parts per million, which is about less than 4%. So I'm breathing out, getting rid of carbon dioxide in my lungs. I get rid of that carbon dioxide, okay? Just like Claudia said, what we've got is we get rid of that carbon dioxide and we pull this all the way over in this direction. So we're pulling it through multiple reactions. If I shift this way, I have to consume hydrogen to do it, and hydrogen also goes down. So that's why in my lungs, the pH in my lungs is about 7.6, pH in my tissues is about 7.2. Depends on how hard I'm working out, right? What those pHs are. Questions? Okay. We also saw it, the example I was headed for was, if you think about DNA, let's say I've got DNA and I've got that 3' hydroxyl. Okay, and I'm going to add, let's make it a CTP to it. Right, what happens? I add the CTP, I keep one, there's only, how many phosphates are between the bases in DNA? One, right. So this is going to be a CMP on the end now, right? I'm not drawing all the atoms here. It would take forever. Okay. But what's the other thing that's left over? I'll draw all the atoms here. You guys remember this guy?
Yeah, that weird thing. It's not stable, right?
I don't know how stable it is, but it gets immediately ripped out. I mean, it's spontaneous. It spontaneously degrades, but you need an enzyme to help it. So in terms of stable and kinetics, I don't know how stable it would be on its own, but there's an enzyme that rips this down immediately to make 2 phosphates. So this is another example of where we have another reaction coupled to help drive this one forward. We're driving forward the attachment of nucleotides to a growing DNA chain by one of our products is pyrophosphate, which immediately gets ripped down to phosphate. So there's very, very low concentration of this. And once again, Le Chatelier's principle, this is low, so it drives it in that direction. Does that make sense? Okay, today is the introduction of a memorization fest, because I've already given you like the cofactors from last time, right? I'm sure you remember them from over break. But now we're going to go talk about glycolysis. All right, so let's see. This is glycolysis right here. I'm going to show it really small, and then I'll blow it up so you can see it, right? So we can see all 10 steps. I'm still working on it. Here, let's try this. There we go, all 10 steps of glycolysis together. Okay, this is glycolysis overall. I should print out a set of these to hand to you so you can look at them while we do go over it for next time. I'll do one for that and gluconeogenesis. which is the reverse process. Well, let's go ahead and make this big so we can see it. Is that big enough so you guys can see it? Looks like it's a little fuzzy based on this. Yes.
For future reference, what of that do you want us to know?
Oh, I'm about to tell you that.
Oh, okay. Draw it from scratch. Right.
Yeah.
We had to do that last
Yes.
Yeah.
Okay. So what you need to do is for glycolysis, know all names of all compounds. Be able to draw all structures from scratch. Okay, know the names of all the enzymes. All right, so this is a flashcards memorization fest thing. You know, I wouldn't do this to you if I didn't feel it was important, and it's important because I've seen it on standardized tests when I recreationally take them or look them over. Okay, so
Just taking the MCATs for the vibes. Right?
We need notes for the quiz, right?
that's kind of a lot for the quiz on Friday, probably. I'll give you a little more, but it'll be on the first test. Okay, so we'll leave it off the quiz, but know the cofactors. Be able to do the cofactor stuff. Okay. All right, so knowing that, let's go ahead and let's take a look at this pathway though, because there are important things here that are things that we might want to make a note of. First off, when we look at this pathway, we don't have the whole thing up, right? There's 10 steps. But what do you see that's different between this first step and the second step there? No ATP, but no equilibrium is what I'm after here, okay? So what we've got here is we've got some steps that have single arrows and some steps that have double arrows. The single arrow steps are spontaneous, really only, or very spontaneous in one direction. So let's make a note of that. Okay. So these steps, so I'm talking about glycolysis right now, but I'm going to, on Wednesday, talk about gluconeogenesis as well, which is the reverse process. So we're going to see this is a catabolic pathway. We're breaking down glucose, and the anabolic pathway is the reverse one. Which of these steps, one or two, are going to be driving this reaction in the catabolic direction? One, right? And if we zoom out, let's see, maybe I'll do this with, make a mess of it if I try and make it bigger and smaller. Let's do it this way. Nope, doesn't work there. Okay, so I make it bigger this way. I don't know if you could see it. Can you see double arrows and single arrows where you are? Okay, so we have how many single arrows do we have? 3. We have a single arrow here at the beginning, we have a single arrow here at step 3, and we have a single arrow down here at step 10, and those reactions are driving the whole process. The rest of these could go either direction. And in fact, when we go and look at gluconeogenesis is only different here, here, and here. Otherwise, it's using the same reactions. Because an enzyme can catalyze a reaction in both directions, right? It lowers, remember, it lowers the activation energy. So if I have a reaction, and this is what that's what my uncatalyzed path looks like. If I then have my catalyzed path looks like this, I have lowered the activation energy in this direction, but I've also lowered it in the opposite direction. Okay? So it's a great example of conservation here for these pathways. For all the things we look at, we're going to see pathways that are very similar okay, and are going to use the same enzymes, assuming they're in the same cellular compartment, which won't always be the case, right? But if they're happening in the same place, they will use the same enzymes for these reactions close to equilibrium, and then they'll have some specific reactions that are very spontaneous. And so for gluconeogenesis, this has to be different that, so it goes the other direction. Does that make sense? Okay. So I lost it. Okay, so they're very spontaneous in one direction. These reactions are great sites for regulation. So you could imagine that, we'll talk in more detail about this on Wednesday, but if I've got two reactions, I've got glycolysis and gluconeogenesis, and one's the catabolic reaction and one's the anabolic reaction, right? You're not going to want both of those running at the same time. You're going to want to either break down glucose or make glucose. And so you would regulate at these steps that are very spontaneous in one direction. If I regulate at this step, what happens? But yeah, if I slow down this enzyme, stop this enzyme from acting, I stop stop glucose from being broken down, and I stop glucose from being made, both. And I'm not going to want to do that. Okay. All right, so they're great sites for regulation. Another thing I want you to note is, let's back up just a little bit. Or make it a little bit bigger or smaller. Okay, we have two phases here. All right, this right here, there's an energy investment phase, and then there's an energy payoff phase. All right, so these first 4 steps, 5 steps here, are energy investment. Looks like that they're defining the first 5 steps here as that. And then the next 5 steps are energy payoff. Really, your energy investment here happens, can you see this? We have an ATP going to ADP. So we're putting in an ATP. We're putting in another ATP here, all right, to get things ready. And then what happens, and this is a little confusing, at this point, let me scoot up just barely and make it bigger. At this step right here, we have fructose 1,6-bisphosphate. We start with glucose, right? 6 carbons in glucose. It's blurry, but 1, 2, 3, 4, 5, 6 carbons in fructose. And then that gets broken up into 2 3-carbon sugars. Okay, glyceraldehyde 3-phosphate and dihydroxyphosphate, or dihydroxyacetone phosphate, sorry. So we end up with these two here. And then what we have is this funny equilibrium arrow, because they're going to both be made. What happens is glyceraldehyde 3-phosphate is what gets taken down. Okay, so we have our glyceraldehyde 3-phosphate, which is this one. We're going to go ahead, and that gets pulled down the rest of the pathway. So what's going to happen since this is an equilibrium? If I pull this out, makes that gets pulled out, makes this gets pulled out. Okay, so it all becomes glyceraldehyde 3-phosphate and runs down the pathway.
So you said glucose gets broken down into 2 what?
2 3-carbon sugars. So we have 1 6-carbon sugar, 2 3-carbon sugars. But what that means is that here they have the 2s, okay? But you might not get a figure that has the 2s here, because it's just showing 1 molecule. Well, here it's showing 2 all the way down. So this is a good figure for that. But sometimes they don't. All right. But what goes on here is now we have 2 of everything from above. We had 1 glucose, makes 2 of each of these 3 carbon sugars that are going to be further broken down. Does that make sense? Okay. So what that means is we put in 2 ATPs up here, right? Down here, we're going to get some ATP back in this step, but we get 2, one for each of the glycerol, one for each of these 2 things, right? And then we're also going to get another ATP at the bottom, right here. But we get 2 because there's 2 of each from 1 glucose. Does that make sense? Okay. So let's go ahead and write that down. So energy investment, 2 ATP in, and energy payoff, 2 ATP out. All right. This is the whopping 2 ATP that you get from glycolysis directly. Is oxygen used in getting that ATP? I've been flying this thing past you, right? So it's not exactly fair. But there's no oxygen in that. The oxygen is later. So what's going to happen is this pyruvate at the end is going to get made into acetyl-CoA that's going to go through the citric acid cycle, and that's going to get fully oxidized, and then we get the electron transport chain. And way at the end of the electron transport chain, I'm just remembering that I must have written on my notes, write out the whole thing, and I didn't. So we'll do it next time. But way at the end of the electron transport chain, that's where the oxygen is. It's electron transport chain. So you get 32 ATP from a glucose, and you only get 2 of them here. Yes?
Wouldn't it be like 4 out? Because if you put 2 in to get 2 out, you would need.
You put, did I say 2 out?
Yeah.
You're right. It's 4 out. Thank you. Okay. But the deal here is that when we did all our lactate dehydrogenase stuff last time, right? Lactate dehydrogenase exists for when you don't have oxygen to get this tiny little bit of 2 ATP out of some more glucose, right? It's this tiny little extra boost. Most of the ATP you get from glucose is involved in this NADH here going down to the electron transport chain, and more NADH that gets made in the citric acid cycle. Okay, from the electron transport chain. And we'll talk more about it next time. You guys have a good